Hello and welcome to this presentation on the nature of SARS CoV-2 variants. This presentation is a follow-up to my lecture on COVID-19, providing more information on how virus variants are generated and how they are established, with particular reference to SARS CoV-2. Now, before I start to talk about SARS CoV-2, I want to introduce you to some genetic aspects of virology that will help you understand better what is actually happening now. This presentation deals with mutagenesis in viruses, viral transmission, the lineages worldwide of SARS CoV-2, the new variant SARS CoV-2 viruses, the mutational changes in the new variants and why it might interfere with their spread and their infectivity, and why so many mutants have been acquired. Finally, I will talk about the South African new variants. Thank you. Let's begin this presentation with some definitions. And the first definition I want to give you is one for mutation. A standard definition for mutation is that it is a change in the hereditary material that is transmitted to the descendants of the organism or virus. Now, there are many types of changes that can happen to hereditary material. At the level of the gene, changes can be made at the base pairing level, where bases may be changed, lost, or inserted. All of these changes may change the reading of the genetic code, which may change the sequence of amino acids in a polypeptide. As well as changing polypeptide sequences, mutations may affect no change whatsoever. These types of mutations are referred to as synonymous mutations. And the reason why we have synonymous mutations is because a codon can code for the same amino acid as another codon. This is referred to as degeneracy. Okay, now let's just assume that the changes we want to make to our codon do affect the change in amino acid residues in a polypeptide. How has this happened? Well, if we look at the little red box in the slide, we can see that we have 4 codons described there, each represented by 3 ribonucleotides, okay? And each one of those give rise to a different amino acid residue. Those I've represented as coloured balls. Okay. Now, if we look at codon 2, we can see that there's been a change from a blue amino acid residue to a green one. Okay. We would write this as blue to green. Blue being the original amino acid residue, 2 the codon position, and green is the new variant present in the amino acid residue. Now, changes in the amino acid residue can have important consequences for the function of the protein or polypeptide in the cell. Most proteins do not function as single polypeptides. Often proteins are accumulations of different polypeptides or even the same polypeptide. So, as an example for that, if we consider the spike protein of SARS CoV-2, then yes, that is produced as a polypeptide, but it doesn't function as a polypeptide. It can only function when there are 3 polypeptides linked together to form a trimer. Okay? So in its functional state, the S polypeptide is a trimer which then can interact with the receptor protein. Now, there are 2 levels at which changes in the amino acid residue can have an influence. The first one, of course, is actually the binding together to form the trimer. This binding is done by interactions between amino acid residues on the polypeptides, and they have to be correct for the correct shape to take place. Second type of change that can happen, that can affect the functioning of the protein, is actually changes in the interactive site. So here we would be interested in changes within the polypeptide that actually influenced the interaction site with the receptor. Now these can be either improved or they can have their efficiency decreased in binding, depending on the type of amino acid change that has taken place. Mutations occur because of errors in replication or because of damage done to the hereditary material. Replication is performed by enzymes known as polymerases, and these can copy nucleic acids very accurately or very poorly. The level at which they make mistakes is known as their fidelity level. Polymerases with a high fidelity often have proofreading abilities that allow correction. Agents that cause damage include ionizing radiation and chemicals that interact with nucleic acids. All organisms have systems that repair damage, and this reduces the level of mutation. Now, the level of mutation is measured as the mutation rate, and this may be defined as the number of mutations per site per generation, or the number of base pair substitutions per site per year. Viruses tend to have a higher mutation rate than bacteria, fungi, plants or animals. And of the viruses, RNA viruses have the highest mutation rate. If you look at the diagram, you can see that whether you're using the mutation rate as mutants per site per generation, then we can see, or whether you're measuring mutation rate as substitutions per site per year, we can see that the single-stranded RNA viruses have the greatest range of mutation rates that vary from 1 in 1,000 to 1 in 100,000. The coronavirus has proofreading ability with its RNA polymerase, which lowers the mutation rate somewhat, and the rate for coronavirus mutation is around about 1 in 10,000. Once a mutation has been established, it can be spread in the population by infecting other hosts. We can sample a population of infected individuals to see what mutation each virus carries. This allows us to follow the spread of any particular mutation in the population. The spread of a mutant variant may be due to chance, or it may be because it is better at causing infection. Let's say because the mutation confers a benefit to the virus so that it is selected. Let's consider random chance first. If the mutation does not confer any selective advantage, and if each mutant variant has an equal chance of replication, then the proportion of mutant variants in a population will reflect the proportion in the foundling sample. This is known as the founder effect or founder principle. For instance, in population 1 in the diagram, the predominant form is the blue variant. This is not because the blue variant bestows an advantage. It is because the blue variant has the greatest proportion in the sampling from the original population. Similarly, in population 2, the yellow and green variants are codominant because they are codominant in the foundling sample. In this way, variants may be fixed or lost from a population, and it is all down to random chance in which variants are actually selected to actually form the new population. If, however, the variant bestows an advantage, then we would not expect this to happen. We would expect the variant to be replicated unequally, like in population 3, where the pink variant dominates, despite being in the smallest proportion in the foundling sample. We have to take care though when describing whether a mutation has a selective advantage, because there are other factors that we have to consider that can cause unequal replication of variants to take place. It is for this reason that usually we look at the rise of mutations in populations, and then we perform laboratory experiments to see if there is a real advantage given by a particular mutation. The primary mechanism of SARS-CoV-2 transmission is by inhaling or having contact with respiratory droplets containing the virus. These can be large droplets or aerosols emitted by an infected person when they cough or sneeze. Infected people can even transmit the virus in this way when they are asymptomatic, when they show no signs of the disease. Because SARS-CoV-2 is transmitted by airborne particles, gatherings of crowds are likely to increase the risk of infection, particularly indoors where ventilation is limited. Furthermore, it only takes one infected person to make any gathering a potential source of infection. We have seen this at so-called super spreader events, where increased levels of infection are observed after such an event occurring. Think of Cheltenham racecourse, the Liverpool match at Easter. All of these were super spreader events where the rate of infection increased dramatically after the events. In addition to this, we can see that the virus is spread by mainly household gatherings. So the likelihood of having a household gathering where just one person comes into the gathering can infect everyone else. There is another area of transmission that we need to consider. Because the receptor for SARS-CoV-2 is also found in the gut, this means that the virus can infect the gut and produce particles, which means that bodily wastes may also be a source of contamination. It's therefore suggested that when you visit the toilet and you flush the toilet, you keep the seat down when you flush, because that will prevent aerosols from entering into the environment. How can we start to classify the different variants or types of SARS-CoV-2? Well, today, with molecular biology techniques, we can sequence the whole viral genome from infected people. We can then compare the sequences from the different infected people to see what differences there are in the nucleic acid sequence. When we do this comparison, we can also make a graph that shows the relationships between the different variants. Such a graph is shown in the slide. When we do this, we find that there are 6 main groups. We call these groups clades, and a clade is just a group of related organisms or viruses that share a common ancestor. The oldest clade in this group is the 19 group, while the 20 clades represent those viruses that have come into existence this year. Each clade is made up of lineages. These are the small coloured circles that we can see in the graph. And each lineage is separated from another by several mutations, usually between 2 and 8. Now, when we look at the different lineages, for each country, we find that the values vary dramatically. China, the origin of the virus, only has 22 lineages, while the USA has 769, roughly about 20% of all known lineages in the world. Now, a part of the reason why this may be related to how each government responded to the pandemic, where some governments enacted a strict shutdown, which would limit travel, which would limit the spread of the virus. If you stop the virus from replicating, then you will stop the number of mutations building, and hence have fewer lineages. But I do not pretend that this is the whole answer. There are other factors that we have to consider when we look at this information. In Europe, we have lineages that belong to all of the main clades, including the ancestral clade, which is clade 19, both parts A and B. These were originally viruses that entered the UK from China. But from then, the virus has grown, it's been introduced around the world, and that has generated the many other types of lineages that we see. Now in Britain, we find that most of these lineages belong to the clade 20A. That's where our greatest diversity of the different viral types exists. So where does the new variant SARS CoV-2 virus belong? Well, if we compare its sequence with all these other sequences, we find that the closest relationships of this virus are in the clade 20B. And they are novel within that group. And they are novel within that group because of the mutational differences that this lineage actually has. Normally, when we compare the lineages between the different viruses, we find the differences in number of mutations that they have varies between perhaps 2 to 6. With the lineage for the new variant, however, the number of mutational differences between this lineage and all the others is somewhere in the region of 23 mutations. This is a huge number of mutations to have actually evolved in that time. Now, the original name of this virus, this new variant virus, was called pangolin lineage B117. That has now been renamed as VUI 2020-12/01. Okay, so if you see the sign VUI 2020-12/01, then that is the new variant, but it is the same as pangolin lineage B117. And it's actually been put into a subgroup all by itself because of these large number of mutational changes. And the new group for that, the subgroup for that, is called 20B-501YV1. Now, why has it been called 501? 501 here is an important number because that refers to the position in the amino acid residue that's changed for the spike protein. If you remember, the spike protein is the protein that interacts with the receptor. And what we find with these mutational changes for the new lineage is that out of all of the 23 mutational changes that have occurred, 8 of them are within the spike protein. So just how unique are the changes that have occurred in the spike protein in the lineage B117? To answer this, a comparison can be made between the sequences of the spike protein from all the other lineages, specifically looking at those that have changed. These are the codon sites 501, 570, 681, 716, 982 and 1118. When we compare these sequences, we can then construct a graph showing the relationship between the lineages and the occurrence of those mutations. Now, the graph that's shown in the slide is the occurrence of the different lineages against time. And we can see that there is only one lineage that has all of the changes there, when that is the lineage B117. But this does not mean that another similar lineage with similar mutations cannot occur. It can, and in fact it has. Just recently we find that a similar variant has emerged in South Africa, which has forced the government to actually close travel, or to place travel restrictions between South Africa and the UK. Now, the problem with letting a virus replicate unabated in a population is you give it the chance to adapt better to the host. And this is what's happening now. The virus is actually evolving, and is actually tuning its attachment to host cells so that they're more efficient. The next slide in this presentation will show you a graph where the emergence of the different lineages is measured against the number of mutational changes. I've included this figure just to show you the number of mutational changes that have occurred in a graphical sense. Up to now we've been looking at diagrams and graphs which have been based on time. This graph is based on mutational differences, and we can see that the B117 lineage far outstrips the number of mutations it has compared to other lineages that we find in this country. The origin of B117 comes from a lineage that is known as B1, and this is the dominant lineage circulating in Europe. It came out of Italy earlier in the year. So why is the transmission rate so important? Well, it's important because if the new variant can spread far easier, it means that it will rapidly infect a large number of people, and it will spread more easily from one location to another. And this potentially can put the NHS at risk, because if we have too many people infected, too many people going to hospital, then the resources we have to look after patients within hospital will suffer. And so it's really important to try to keep the number of people entering hospital with COVID-19 down as small as possible. With a new variant, of course, spreading rapidly like this becomes more difficult, and it becomes more difficult to control the virus. A faster spreading virus is not going to be so easily controlled by mild lockdown conditions. It's going to need something far more dramatic and draconian. When a new variant of a disease-causing virus appears during a pandemic, public health scientists ask three basic questions that help them determine what advice they should give to keep the public safe. These questions are: number one, Is viral transmission more efficient with the new variant? Number 2, are the disease effects greater with the new variant or not? And number 3, is the new variant sensitive to current vaccines? At present, we can only answer the first question with some evidence, although the drug company Pfizer has said that the vaccine that they produce will actually countermand the new variant, will interact with the new variant. Okay, so let's look at the evidence that transmission is greater with the new variant B117. The 3 maps of Britain shown in the slide and the histogram along the bottom indicate that the new B117 variant has a much greater transmission than other lineages. The first map on the left shows the distribution of 3 other lineages, B117, B1170 and B1174. Now both of these other lineages, B170 and B1174, were thought to be possible candidates that show greater spread. But in actual fact, when we look at the distribution of these lineages within the UK, we can see that they are primarily localized. The lineage B1174 was thought to actually have spread from Scotland, and we can see that there are cases of it down in the southeast. However, when we compare the spread of these lineages with B117, we can see that the spread of B117 is far, far greater. And that is shown in the histograms below, where we can see that the rate of infection caused by B117 is clearly a lot greater, and that it is spread more quickly. This can be seen in the 2 middle maps that show the distribution of B117 before November the 29th and after November the 29th. And here we can see that the distribution of B117 was at first in London and in Kent, and that has rapidly spread into Sussex and many other counties within the southeast. We can also see, if we compare that to the original map that I talked about on the far left, that now B117 has literally spread all over the country. So, and it has done this very, very rapidly. Although we wait for lab experiments to confirm that B117 is more efficient at transmission, it is really unusual that such a rapid spread of a particular virus is unrelated to the efficiency with which it infects its host cell. In the previous slide, we have seen that the lineage B117, the viruses have a greater transmission. But why do they have this greater transmission? The answer to this lies within the nature of the mutational changes that have occurred within this lineage. The B117 lineage has 23 mutations compared to other lineages. 6 of these make no difference whatsoever. They are called synonymous mutations because they make no change. The other mutations occur in genes OFR1A and the N genes. But the mutations that are of greatest interest are those that have happened in the spike gene. It is these mutations that are important because they can improve or decrease the efficiency with which the virus enters its host cell. One mutant that has allowed this to happen occurs at ribonucleotide position 23063. Here, a thymidine replaces an adenine in codon 501. And this results in a change in the amino acid sequence from an asparagine to a tyrosine. The position of this mutational change can be seen in the 3D image of the spike protein. Here, the spike protein is represented as a trimer, and we can see that the 501 location is directly in the center where the receptor protein fits. This simple change increases the binding of the spike protein to the ACE2 receptor. So the virus, this particular virus, is more effective and more efficient at actually binding to the host cell receptor. The second important mutation is the one that occurs at ribonucleotide position 23604. This mutated change creates an additional furin cleavage site necessary for the spike protein to be cleaved so that it can enter the host cell. So this mutation improves viral entry into the cell. So just these two mutations alone allow for a greater transmission. But within this lineage, we have a third set of mutations. Now, these are not base pair substitutions, they are deletions. In other words, a region of the gene has been deleted. Okay, now these deletions occur at positions 21765 and 21991. These deletions cause a section of the spike protein to be removed. Now, you might think that is a good thing, and often deletions cause for the inactivation of the protein. Unfortunately, not in this case. The region that's been deleted in this case is a region that is the target for antibody 484. So the antibody cannot bind to the spike protein as it would normally do. This allows the virus to escape, or at least part of, the host's immunity system. So in actual fact, with these three mutations, what we have is a mutation that increases the binding of the virus to the host cell receptor. It allows a greater efficiency of entry of the virus into the host cell, and it allows the virus to go undetected by part of the immunity system. These three things alone would suggest the reason why this particular lineage has spread so rapidly throughout the country. Now, earlier on I said that Pfizer have said that they expect their vaccine to raise antibodies to this particular variant. What is their confidence? Why are they so confident that this is the case when we can see that mutations carried in this lineage actually help the virus avoid the immunity system? Well, the reason for their confidence is that their vaccine targets a different region of the spike protein. So in all honesty, they would really expect that still to work against this particular variant. So a very good reason to become inoculated against this virus. One important question remains about the evolution of lineage B117, and this is, how did B117 acquire so many mutations in such a short time? Is it because it can evolve faster than the other lineages? To answer this question, we have to consider how mutations can accumulate and be introduced into new lines. Now, a lineage can start off with a single mutation and over a series of replication events acquire other mutations. But this is a relatively slow process. It can be speeded up if the virus can exchange genetic information with other virus lineages. And it can also acquire more mutations if the infective cycle is prolonged, i.e. if it lasts longer than usual, producing a greater number of daughter progeny viruses. In answering this question, we first have to establish whether B117 has a greater rate of mutation than any of the other lineages. We can do this by taking the regression of mutant changes against time for the B117 lineage and all the other lineages independently. A graph showing this is shown in the slide to the right. We can see from these regressions that the rate of evolution in B117 is very similar to that of other lineages. So in conclusion here, B117 is not evolving faster. There must be another explanation. Part of the answer to this comes from the observation that B117 has a greater genetic divergence than the other lineages. We have already seen this in graphs like the one shown on the left in the slide, where we can see that lineage B117 has a far greater number of mutations accumulated, so the diversity within all the viruses within this lineage is actually far greater. What this indicates is that there must have been a time during the evolution of this viral lineage where it had undergone a period of accelerated evolution, either by being able to recombine or having a less restricted environment to grow in. By that I mean it is allowed to extend its replication cycle to produce more viral particles per infection and therefore a greater number of mutations. Okay, so if we're looking for a less restricted environment, where could this be? The answer is in the following slide. A suitable unrestricted environment where the virus can replicate is immunocompromised patients. And this was suggested by Victoria Avanzato and her colleagues, who was working with immunocompromised patients and observed that one of their patients, who was infected with SARS CoV-2 but asymptomatically, continued to shed the virus after 70 days after infection and continued to shed viral RNA up to 105 days after infection. So this clearly indicates that in this particular patient, the viral infective cycle was extended, and this increased infectious cycle allowed the creation of a large number of mutant varieties to come into existence, and that these could possibly interact with each other to create even more novel lineages. So an extended infection in an immunocompromised patient could give rise to the accelerated evolution that we observe in lineage B117. Now, I should point out at this stage that this is just a theory, but it provides a very good hypothesis that we can test. If it is true, then it makes it of even greater importance to protect patients in hospitals from being infected with SARS CoV-2. While we have been occupied with lineage B117 in the UK, another variant lineage carrying a mutation in codon 501 of the spike protein has emerged in South Africa. This lineage, B1351, contains a range of different mutations in addition to the one in 501 that we find in B117. So this is a very different lineage. The new lineage has spread rapidly throughout South Africa, and it has been given its own subgrouping, which is 20C/501Y V2. So now the new lineage is not within the B2 cluster that B1117 came from, it is within the 20C cluster. Now this is quite disturbing. The graph presented on this slide shows the relationship of different B1351 lineages in South Africa. This lineage has viruses that contain the N501Y change, but also contain a change at codon position 484. This is also within the receptor binding region of the spike protein, and it is assumed that this helps to improve the binding once more of the virus to this receptor protein. The lineage, however, does not have the deletion mutations of lineage B117, but it has been reported that viruses of this lineage have a greater viral load on diagnostic swabs than other lineages, and this may make it more transmissible via aerosols. Unfortunately, this lineage, the B1351 lineage, has already entered the United Kingdom. We are approaching a very dangerous period of this epidemic, where swift action is needed to actually stop the spread of this virus. Failure to do this will lead in a far greater number of people dying. I've included this last slide to show the transmission of the variants in South Africa that have the mutations in the 501 site, as well as mutations in the 417 and 484 sites. And we can see when we're looking at this rate of transmission that virus types that have a specific NKY type for sites 417, 484 and 501 have spread and become the dominant forms within South Africa. So this just adds further evidence that this particular virus variant, the B1351 variant, actually spreads, has a greater transmission than the other forms that are present. Well, I hope you've enjoyed listening to this. I do not mean to depress people unduly. We have a vaccine on the way. We have a number of vaccines on the way, which will help control this virus over the course of next year. And I'm sure that this will happen. So with that in mind, I wish you all a happy new and prosperous new year.